Hi guys, so today we're starting by going over a couple of monohybrid crosses before we move on and add the layer of difficulty of the dihybrid cross. So the monohybrid cross, or the one factor cross, remember, is looking at just a single characteristic like seed color or seed shape or other things we've talked about in our problems was like the arm length of on a saguaro cactus, or whether or not a person had a widow's peak. So all of these are monohybrid or one factor crosses. They look at just one characteristic and how it changes. So in this example, we're just going to do two examples together. In this example here, we are starting in the F1 generation. So in our F1 generation, meaning that these two seeds, these smooth seed plants, they can either have smooth seeds or they can have wrinkled seeds. And in the F1 generation, when we cross a smooth seeded plant with a wrinkled seeded plant, we get all smooth seeds. And so the first question is, what does that tell you about the P generation or the parental generation? The parental generation needs to be one true breeding smooth seed with one true breeding wrinkled seed. So that's kind of what we're getting from this first cross. What is dominant? Well, whatever factor shows up in our F1 generation, that is the dominant phenotype. So we expect that dominant is smooth. And so that means that wrinkled is going to be recessive. So we have now two heterozygous smooth plants. And so the question is, what will we see in the F2 generation? So let's take a look. So this is the first bit of information that I have put down, and this is being taken right here from this part of the example. It shows us that both of our individuals are the heterozygotes, big S, little s. And remember, I've mentioned that S maybe isn't the best one to use, but in this case, I'm using an S, a big S that's twice the size of my small s in order to keep things nice and simple. So now all we need to do is the Punnett square for the heterozygote cross. So we make our square and we put in our big S, little s, and big S, little s. And then it's time to fill this in as we go across. So in my first box, top left, I have big S, big S. And then next to that, to the right, I have big S, little s. And you can see that I'm keeping my big and littles very clear so that I don't confuse myself when it's time to interpret these results. In the lower left-hand square, I have big S, little s again. And then finally in my last box, two small s's. And so I expect in my F2 generation to have the genotypic ratio of 1 to 2 to 1. And remember, that's the homozygous dominant to the heterozygote to the homozygous recessive. For my phenotypic ratio, I expect to see a 3 to 1 ratio, smooth to wrinkled. And so keep in mind that one of the things that you need to pay attention to when you're answering a genetics question is, did it ask you for the genotype or the phenotype? And careful reading is always going to be the key to those kinds of questions. So make sure you're always doing your careful reading to make sure that you're getting maximum points. All right, good job on that quick review of monohybrid crosses. So here you can see the rest of that diagram, and we see that we did a good job following through with our big S and our little s and distributing that, and then we end up seeing here the genotypic ratio: 1/4 big S, 1/2 big S, small s, 1/4 small s, or we could call that a 1 to 2 here to 1 genotypic frequency. And then we also see our phenotypic frequency, which is 3 smooth seeds to the 1 wrinkled seed. So we can also call that 3/4 smooth to 1/4 wrinkled. And you actually can answer either way. If it's multiple choice, you should pick the way that it's set up in the multiple choice. But if you're writing out the answers, which for some of them you will be, you can answer either in a 1 to 2 to 1 way, or you can say 1/4, 1/2, 1/4. Either one is acceptable because they're both mathematically accurate. The next thing that we want to talk about here is the idea of the test cross. And so it is fairly common that we would see a dominant phenotype individual, and we wouldn't know if they were a heterozygote or homozygous dominant. So let's look at that and make sure we understand what we're saying. So what I'm saying is that it's pretty common to see something with its dominant phenotype. So maybe the dominant phenotype is a white sheep color. Okay, so in sheep, the dominant phenotype is a white sheep color. So if my dominant phenotype is white fur, if I see an individual who has white fur, I don't know if they are white and white, homozygous dominant, or if they're white and the recessive type, black sheep, black fur. So if this one is white and this one is white, I don't know which one it is by looking at it. So the way to figure it out is to do a test cross. So with a test cross, you're going to cross against a homozygous recessive. So this would be a black sheep with black fur. So we don't know which one it is, whether it is the heterozygote or the homozygous dominant. So we're going to test it against the recessive phenotype, in this case, black sheep. So we're going to go ahead and look and see what that would look like. So when we do this kind of a cross, what we're really checking for is this W true, or is that W true? And so that's really what we're looking for. And so when we do this cross, we can tell from the offspring. So my white sheep is either this genotype or this genotype. And so what we're going to do is we're going to predict the frequencies of their offspring. So no matter the cross, this information stays the same. We're always getting the 2 small W's from our test cross. We're always getting the big W from our unknown individual. The question is whether it's that or whether it's this smaller W. And we will find out when we look at the frequency of the offspring. So as we do our Punnett square, what we see is half of the Punnett square looks exactly the same. Okay, this top part. What changes is whether on our bottom row we have heterozygotes or homozygous dominant. So if we look at this test cross, what we see is in this cross, from a white sheep with a black sheep, we're going to get 100% white fur. And in this cross, we're going to get 50% with white fur and 50% with black fur. So what we see is that in our offspring, by looking at the offspring of this cross, we can work backwards to determine the genotype of the parents. So if we were to do this cross, perform the cross, and we were to see that we had 50 offspring and they all had white fur, that would tell us that it was this genotype up here. If we had a setup like this, we did the cross, we performed the cross, and we saw 10 white sheep and 3 black sheep, that would still point to this cross. That's not a 50-50 split. However, if the adult was big W, if the unknown was homozygous dominant, we would see 0, no black furred sheep. So even 1 will tell us that we are looking at a test with a heterozygote. As we continue through the different genetics problems, this idea of the test cross and basically the skill of working backwards from what did the offspring look like, what were the phenotypic and frequencies, and you take those backwards to predict the genotype of the previous generation. And that's actually one of the really fun kinds of questions that you will definitely see throughout this genetics practice. So now that we've wrapped up this concept of the monohybrid cross, we're going to be taking it one step further, and we're going to start studying two characteristics at the same time. We call that a dihybrid cross, or a two-factor cross. This is going to look at two different variations at the same time. So we might look at seed color and seed shape. Is it yellow or green? Is it round or wrinkled? If they're inherited together, we would see a consistent, if it's yellow, then it's round, if it's green, then it's wrinkled connection. But we're not talking about characteristics that are linked like that. We're talking about independently assorting characteristics, which means that it makes no difference whether this top set of chromosomes goes red to the left and blue to the right, or if it goes blue to the left and red to the right. It doesn't matter. So we sometimes get a red and red combination. We get blue with blue, we can get red with blue, we can get blue with red. All of our different characteristics independently assort. And so we're looking at two separate characteristics, typically on two separate chromosomes that are moving through this process, and we're keeping track of them together. And obviously keeping track of two things is more difficult than keeping track of one thing. If you don't believe me, babysit. So to start out this problem, we need to start with some factual information. So we're looking at the facts, and we see that yellow is dominant to green in seed color, and that round is dominant to wrinkled. And so the first thing that we need to do is we need to identify the different phenotypes that we're going to be seeing and their corresponding genotypes. So let's get started with that. So we're starting out with our genotypes and phenotypes corresponded to one another. So yellow, big Y, little y, green is small y, because that's the recessive. Round is big R, blank, and wrinkled, small r, small r. So now we're going to start out with true breeding yellow round seed with a true breeding green wrinkled seed. And so what we see is true breeding yellow means homozygous, right? A true breeding yellow round is crossed with a true breeding green wrinkled. Green wrinkled. So first off, when we do this cross, we can take a shortcut with our first Punnett square, okay? The offspring from this mating is going to get one of each type of allele. So from this parent, we will contribute a big Y. From this parent, we contribute a small y. From this parent, we contribute a big R. From this parent, a small r. So 100% of my offspring in my F1 generation, so this is my P generation, here's my F1 generation, they're going to be yellow and round, and they're going to be heterozygotes. So that's what it looks like to do a P true breeding to F1 generation. In our next generation, we want to take this offspring and see what happens when they cross. So we have our heterozygotes crossing. Now the first thing we need to do is determine what our possible allele combinations are. And to do this, you basically FOIL. So we're going to go, we could get a big Y with a big R. We could get a big Y with a small r. We could get a small y with a big R. And we could get a small y with a small r. So these four alleles are our possibilities. And these become the top of my Punnett square. And the bottom along the side repeats. Big Y, little y, little y. Take up all the space I have here. Big R, little r, big R, little r. And I'm just following the pattern. Big Y, big R, big Y, little y, big R, little y, little r. I'm following the pattern. And now I want to make this a filled in square. So some of you maybe know what's coming. We've got to fill this all in. So I'm going to take a second and fill my Punnett square all in while you take a second and fill your Punnett square all in, and we will meet back. Now I prefer to do it one allele at a time, so I've just filled in my Y's and now I'm going to go back and fill in my R's. So remember, I'm just taking the combination. So this will be big R, big R, big R, little r, big R, big R, big R, little r. And I just keep going along the line, lining them up. The big letter can always go first. So big R, little r. So we have now made our Punnett square, and great job with that. Good job getting your Punnett square together. And now what we want to do is start thinking about, what does any of this tell us about the phenotypes of our offspring? So remember that big Y blank is going to give us yellow, and two little y's give us green. And also remember that big R blank is going to give us round, and little r is going to give us. So what we're going to be doing now is identifying the phenotypes of our offspring. So when we do this, we take and we look in here and we say, okay, big Y, that means that I have a yellow. So my offspring are going to fall into one of four categories. I am going to have yellow round offspring, and they're going to be big Y blank, big R blank. I'm going to have yellow wrinkled, which will be big Y blank and two little r's. I'm going to have some that are green and round. And lastly, I'm going to have some that are green and wrinkled. So what I'm going to be doing now is going through my Punnett square and keeping track of what phenotypes I'm seeing. So if I start here, as I go down the line, big Y's and big R's. This one is yellow round. This one is yellow and round. This one is yellow and round. And this one is yellow and round. Everything in the first column has the same phenotype. Okay, let's scoot over. Second column, first row, yellow and round. Second row, this is yellow, but now we have two small r's. That makes us yellow and wrinkled. Next one, this one's yellow and round again. And yellow and wrinkled. Remember, the two small r's tell me that it's wrinkled. Next row, yellow and round. Yellow and round. Green and round. That's our first green and round. So I'm going to grab another color for my green and round here. Green and round again. And my last column, up top, once again, yellow and round. Yellow and wrinkled. Green and round. And last but not least, my very last one, the homozygous recessive for both, green and wrinkled. So this big 16 square Punnett square boils down to this very simple bit of information. The end result of my dihybrid heterozygote cross is nine individuals that are dominant for both phenotypes. 3 individuals that are dominant for one phenotype but recessive for the other. And another 3 individuals that I describe as being recessive for one but dominant for the other, meaning that they have switched which one they're dominant for. The first one is dominant for the color, but the second one is dominant for the shape. So they've reversed positions, which one they're dominant for. And lastly, a single individual that is recessive for both characteristics. Now, the beauty of this Punnett square, this huge problem, this giant amount of time that we've put in here, is that you don't need to do this every time. This 9 to 3 to 3 to 1 is always the result of a heterozygote dihybrid cross. We won't do another huge Punnett square like this. I won't ask you for a Punnett square with this many squares in it. What I will ask you to do is to know that instead of doing this Punnett square, to memorize the ratio that the results of a heterozygote dihybrid cross are 9 to 3 to 3 to 1. So when you do a heterozygote dihybrid cross, you're always going to get that 9 to 3 to 3 to 1 ratio, where 9 represents dominant for both characteristics. 3 and 3 are each dominant for one of the characters but recessive for the other one, and they trade off. And then the one individual is recessive for both alleles. So this ratio of 9 to 3 to 3 to 1 can be used to predict the outcome of any heterozygote dihybrid cross. Other dihybrid crosses, you can use logic to eliminate some of the columns or rows on the Punnett square. So for example, if you're doing homozygous dominant with a homozygous recessive cross, even though there's all of those characteristics, you really only have one possible allele from each parent. And so you really only have one offspring type for your offspring. If you were doing different variations on the cross, then you can pare down the different alleles that will be produced. So you always want to start out with that foil part where you figure out how many alleles there are, and then you're going to put those as the columns and the rows of your Punnett square. You should not need to do anything bigger than a 4x2 Punnett square yourself. Beyond that, the Punnett square should give you the ratio of 9 to 3 to 3 to 1, in which case you wouldn't do the Punnett square, you would just have memorized the ratio. Okay, so monohybrid crosses and dihybrid crosses are now complete in terms of me giving you information. We will practice more dihybrid crosses in class tomorrow, and then we're going to look at some non-Mendelian genetics when complete dominance doesn't really work out, and instead we have maybe many alleles or a shared dominance. We're going to look at kind of some exceptions and real life examples of how genetics plays out in the animals and plants that we have studied. Good work today, and I will see you in class.